Good day, everyone. Welcome to day 2 of the 2-day Stanford conference. I'm Brian Sparks, Senior Director of Worldwide Operations for the HPC Advisory Council, helping put on this conference with Stanford. With me, as always, is Steve Jones, Director of the Stanford University's High Performance Computing Center. Steve, how are you doing today?
I'm doing well. How are you, Brian?
Excellent. I know you want to talk about some upcoming classes and courses. Why don't we go ahead and start with that before moving on with the 3-hour agenda that we have today?
Yeah, sure. Great. And of course, these courses were developed over the years in partnership with the Advisory Council through our longstanding relationship. So this summer, we're launching a new class in continuing studies. So we've been offering courses through the regular university curriculum and summer session. But this year, we decided to open one up to the general public in the continuing studies format. That's CS74, an introduction to building high performance computing systems. So that will be starting in July, an evening class. It's in an online format, but you'll have direct access to hardware here at Stanford, where you'll learn how to build your own HPC cluster. It'll be based on OpenHPC. We're going to have guest speaker Jeremy Seidel from Intel helping out with that. And I just found out that we're going to include a session on singularity too. So there'll be some cool container things on that. And then we'll be doing some things with our summer seminar series this year in a public format. So you can either visit our website, join our meetup group, or go to the HPCAI Advisory Council website for more info on that.
Great. Thank you, Steve.
Yeah, sure.
And welcome, everyone. And before we begin, just to remind you, if you do have questions for our speakers today, you can either use the Q&A or the chat fields, and our speakers will answer them right at the end of the presentation. So with that, let me welcome everyone to our first presenter, Brian van Essen, who is an information group leader and computer scientist at the Center for Applied Scientific Computing at Lawrence Livermore National Lab. Today he's going to be talking about a new generative model that LLNL created to help enable COVID researchers to produce novel compounds for the disease, and how they were able to reduce that model training time from an amazing 1 day to just 23 minutes. Brian, welcome.
All right. Thank you very much for the opportunity to speak with your audience here. I really appreciate it. We actually have a long-standing, good collaborative relationship with Stanford. As a national lab, we love working with your undergraduate and graduate students as well. I would actually say that Tim Moon is an alumni of Stanford, and some of the research that we're using in this work actually built on stuff that came out of your university. So please, going forward, just keep that in mind. So I have the great privilege today to talk to you about work that my team and I did this past supercomputing SC20, where we were developing new methods for developing scalable generative neural network models that are targeting the creation and development of small molecule drugs to target COVID-19. This was one of several Gordon Bell finalists this past year. And it is a great deal of fun and an opportunity to advance the state of the art in both computer science, research, scalable machine learning, as well as computational chemistry and biology. One of the fantastic things about working at the National Lab is the opportunity to work with a wide multidisciplinary team. These are my colleagues who were direct contributors to this project, but the actual pyramid of who built the capabilities that we're talking about today is actually much taller and deeper. This work was funded by both the ECP ExaLearn project as well as the ATOM institutional consortium, as well as the ASC research program within the National Nuclear Security Administration. So let me again thank them for all of their great contributions to making this work happen. And again, this team ranges from computer scientists to computational chemists, bioinformaticists, and HPC expertise. So what are we trying to do here? Well, drug design is costly in both time and effort. This is the standard pipeline from when you say, let's develop a new drug all the way to let's get it through FDA review. Normally, this is a 15-year process. We've seen amazing heroics this past year during the pandemic, where we got multiple vaccines developed in about a year, which is practically unprecedented. But this work is really focusing on the first part. Sorry, a significant portion of these actually fail in these stage 3 clinical trials. So the work I'm talking about to you today is focusing on these first 2 stages. And the question we put forth is, can machine learning on high performance computing accelerate the time to develop and discover novel initial compounds for these kinds of clinical trials, and really sort of accelerate this entire loop. So what research are we working on? Well, researchers across the community are developing new vaccines that you've heard about in the news, ways to use therapeutic antibodies, but this work is focusing on small molecule antivirals. And the reason that we need to push forward on multiple fronts is that no approach is going to work for every person all of the time. And so you need multiple avenues of attacking not just this pandemic, but future ones that may come, or future ones as well. So within this work, there are really several different phases of drug development. There's hit identification, where you say, this is a chemical compound that might be interesting. Let's refine it and turn it to a lead compound and generate that one. And then the last part of this loop is the lead optimization, where you say, I have a chemical compound that I like, I've curated it from a larger pool, and I want to actually optimize that further. Maybe we want it to be more drug-like, we want to increase its pharmacological effects, minimize risks, but we're at that part of this drug discovery loop. One of the central theses of the computer science and scalable machine learning part of this is, can we increase the swath of chemical space that we're using when we do this lead generation to increase diversity and robustness of the solution? Now, many people have seen these different slides here. Here's a picture of the SARS-CoV-2 protein with its spikes on top. And really what we're looking at, these receptor binding domains here shown in the circle. And if you visualize it differently, the idea is that we can create these small molecules shown here in this blow-up box that get into one of these binding pockets and inhibit the virus from attacking the human. In particular, there are 4 key protein targets that the community and our ADAM team have been working on. And the idea is that you want to screen millions of drug compounds to find good candidates for ways to stop both viral entry and viral replication. Once you find promising compounds, you want to create novel drugs that can actually inhibit this. But the drug design space is on the order of 10 to the 60th chemical compounds are possible. And that's just way too much to have our graduate students and postdocs and research technicians going through manually. And so really what we want to do is, can we accelerate that out of the traditional wet lab design into a computationally driven approach? So the idea is, how can we increase the diversity of chemical space when we're designing this novel drugs? Well, prior work that the ADAM team has been using, and a lot of the community works with millions to maybe tens of millions of chemical compounds to train generative models. There's a particularly successful one called the junction tree variational autoencoder out there in the community. That's sort of the workhorse of the design loop right now. And that's great, except it's slow. And I'll talk a little bit more about that in a few minutes. So what we wanted to do is, we said, well, we want to trade with a much larger data set, 1.613 billion compounds as of the fall of 2020. The data set's now 14 billion compounds, so we're actually tackling that as we speak. But the idea is that we want to increase the chemical space. So we're using 1.36 billion chemicals from the enamine real data set. These are supposed to be synthesizable drug-like compounds, as well as some historical compounds, and a 1 million compound library that is it is indicative that it's going to be more effective against SARS-CoV-2. And so we also have a held out test set of another 2 million compounds. So The idea, so at the heart of this molecular design loop that I'll show on the next slide, but at the heart of this is a molecular generator shown here by the center box, where you have an encoder-decoder pair. And actually, and this is what we're going to be training at scale with leadership class computing and a billion compounds. We also have a molecular optimizer that's going to allow us to perturb where we land in the latent space of that molecular generator. So the idea is you start with a chemical space. On the left-hand side, you take a lead compound, you project it into latent space. And from there, you're going to optimize that latent characteristic for some features you like. And then you're going to produce a new chemical compound based on that latent representation. Now, the thesis of this work, as I said, is if we work with a larger chemical space, can we teach the generator to create new compounds that are more novel, more diverse from what the chemistry community is already working with in their existing sort of corpora of data? And so really that is can we increase the diversity? Okay, so how does this fit into a broader loop? As I said, there's this generative molecular design loop where we have a working compound library. We're going to evaluate and screen these compounds in this top box here. And that's going to use actually a variety of machine learning and traditional data science approaches. We're going to select some based on design criteria, and then we're going to push those through this molecular generator, optimize them in latent space, and add those back to the working compound libraries. We seed this with an initial sort of classical screening of about 700 million compounds where we do molecular docking, rescoring, and molecular dynamic simulations to see the working compound library. And this is the sort of framework that we're fitting into. And again, the work I'm focusing on that was part of the Gordon Bell submission was the this bottom box here was the training of this molecular generator. Okay, as I said, the state of the art is a junction tree variational autoencoder. It's out there in the community. It does a great job, but it's really slow to train. It took one of our computational chemists over 24 hours to train it on a 1 million chemical compound library using PyTorch. So they worked with our computer science and scalable machine learning team, say, how can we make this better? So we wanted to find a new scalable machine neural network architecture, and we're doing this based on character-based sequence models. So the input to this is going to be a smile string. So that's shown over on the right-hand side, the sequence of C's and N's and equals and parens and whatnot. And that just represents sort of this 2D, the original chemical compound. As I said, we're going to perturb the chemical's representation in latent space, and then we're going to decode it into a new chemical, and then we're going to validate our results using something called the Tanimoto distance or Tanimoto similarity metric, which is a way the community used to score chemical similarity, as shown here in the bottom right-hand corner. So where did we start? Well, we stepped back from the junction tree variational autoencoder, and this is a bit of a primer here, but autoencoders are designed to take a given input sequence and create an output sequence. This is not a visual autoencoder, so it's not using convolutional layers. It's using it's a sequence-based one, so it's using gated recurrent units. And so the basic task, again, is create a model that can exactly reproduce what you gave it. So that's great for a bootstrap, but it doesn't do what you need for generative models. You can't hallucinate new compounds. So in the community, there is a character-based variational autoencoder that preceded the junction tree variational autoencoder, is faster to execute, but not as robust as the junction tree variational autoencoder. And we worked with that for a long time, trying to identify whether or not, if by increasing the scale of the data set and the model, we could achieve better performance across these different axes. It turns, and what the variational autoencoder does is it introduces this regularization term in the latent space to be able to allow you to generate new compounds. And so now what you can do is you can start from latent space, put a vector in there and create something new, and you'll get, with high probability, a legal chemical string. What it doesn't do well is it doesn't actually do the lead optimization that we need. It doesn't work terribly well if you put in an input compound that you want, perturb it from that point in latent space and regenerate it. It's just not very robust there. It's very brittle. So we took our knowledge of machine learning and we coupled Wasserstein-based learning techniques from the rest of the community and applied it to this this character-based Wasserstein autoencoder and created a novel model, the character-based Wasserstein autoencoder, that really balances the generative nature of the variational with the ability to ensure better reconstruction that was critical for the latent optimization techniques that we needed. And so it adds this discriminator in the latent space that allows you to actually enforce that the model or guide the model to create better direct reconstruction. And that turned out to be incredibly successful for lead optimization. So what do I mean by incredibly successful? Well, here we are comparing the junction tree variational autoencoder trained on 1 million compounds with our Wasserstein autoencoder trained on 1.6 billion compounds. I'll talk about how much faster it was in the remaining half of this of the talk, but what I want to highlight here is that we were able to get a very comparable percent validity. So some of the times our Wasserstein autoencoder will produce chemicals that aren't actually valid, and so you just throw those away. But the average Tanimoto distance is in some ways far better than what the junction tree variational autoencoder means. And so what that means is when you take your lead compound and project it through sort of the cone of chemical space that you're in is more tightly bound than the junction tree variational autoencoder. So that was a really big win, and our domain scientists were really excited about that. So that's the sort of the science of the model we trained. Trained on 1 billion compounds. And I will say at this point, the main engineering hurdle is to take the model that we've trained at scale and export it to the GMD loop that we showed earlier so that it can be used for day-to-day inference into that design loop. Okay, so to do this, we couldn't just take normal PyTorch or TensorFlow. We needed something that was designed to scale up on leadership class computing. So we've been investing at Lawrence Livermore for a number of years, about 7, on a scalable deep learning toolkit called LBANN, the Livermore Big Artificial Neural Network Toolkit, because I'm terrible at naming things. But it is designed from the ground up to be a distributed training algorithm. It's a mixture of C++, MPI, CUDA, Cudian, and Nickel, the whole alphabet soup. It's built on 3 key libraries: Hydrogen, which is a distributed linear algebra library. It's a GPU accelerated version of the Elemental library created by Jack Paulson. So thank you very much for your contributions there. We really enjoyed. We've created another library called dihydrogen, which is a GPU accelerated distributed tensor algebra library, as well as a high performance GPU communication library that really lets us efficiently in-queue communication and computation when going to GPU resources. And I should say that all this work is open source. It's on our GitHub pages, and we are very happy to work with collaborators. The front end of this is a PyTorch-like model, but it's not the same as PyTorch. You don't typically run this interactively. You don't interrogate your weights as you're going along. This is designed for large leadership class systems. It can run on your MacBook Pro, but that's not really its bread and butter design spot. Just to give you an idea of what this is really it's designed to compose multiple levels of parallelism, model data, and ensemble parallelism effectively when training on these massive data sets. It also has some tips, some tricks in there to be able to scale up. How do you handle this massive I/O and other things? And just as a brief representation here, the idea is that this is showing 2 trainers, each of which is training an instance of the neural network model that they will combine later. But those trainers are spread over multiple compute resources, multiple ranks, multiple GPUs on multiple nodes with parallel I/O, data, and model parallelism within each trainer. So one of the things that was critical to getting to a single trained model on a massive data set on a leadership class system, you can't just scale the mini batch size up to 1 million. So you have to do other techniques. And we've been developing a tournament algorithm called LTFB that allows you to train multiple instances of the model, periodically couple them through a tournament exchange, and then allow a winning model to have seen enough of a tour of the data to be well generalized across the entire data set or the entire test set. So here we have 4 trainers, each of which has model and data parallelism internally. They have their own sets of weights, and they are looking at their own silos of data. They're going to train independently, and within each trainer, they're going to have collective communications, all reduces, and all sorts of things to make sure it's coherently trained. Then at some point, you're going to take the weights of your model and you're going to exchange them with some other trainer. And we do this in a point-to-point manner. So it's actually very scalable when you go up to 16,000 GPUs and 4,000 compute nodes. So you give your model to somebody else. Everybody runs local private tournaments and they pick the winning model to propagate. So sort of like virus replication, and they train the winning model from then on. This allows the strongest models to percolate around the entire set of trainers, see enough of the data that when we do final evaluations, they're shown to actually to generalize really quite well. And one of the key things here is that this method is really scalable. So within each trainer, we have collective communication algorithms with both nickel and MPI. Between trainers, we have point-to-point MPI exchanges so that we can actually achieve that level of scale. Okay, the other thing I want to say about sort of the infrastructure is that when you get to working with 1 billion compounds, you need to be clever and avoid killing your parallel file system. So we developed an in-memory data store that is distributed across all the trainers, and that allows you to ingest the data once at startup time and then never go back to parallel file system. All the data movement within a trainer is kept within that trainer using, again, asynchronous communication algorithms. Okay, so for the Gordon Bell, this was really about how can we tune the performance of this training. We have multiple levels from single GPU performance per mini batch size and performance focus there. How many GPUs do you put per trainer when you over decompose your problem? And how many trainers are you using in this approach? And so the biggest challenge we have is that this character-based Wasserstein autoencoder is actually very small, 27 megabytes of weights. So it's not a huge model. It does not lend itself well to traditional strong scaling techniques. We were able to get some, but the bottom line is it's small. We did use mixed precision tensor core operations. We had to actually keep everything in 32-bit precision and push it into 16-bit precision when we went into the tensor core. So all the numbers you see are mixed precision, but it's not as aggressively in FP16 as we wanted to, primarily because when we trained on the entire data set, the model actually did not converge. On small swaths of the data set, it was okay, but with the whole data set, it didn't actually manage to converge. So there are some techniques we're going to look at in the future to do this. The other thing is that the model is kernel launch limited once you get everything nicely tuned inside. And so there was a huge win by using CUDA graphs over the stacked GRUs calls into the cuDNN library that got us a 1.5x speed up when we did all this sort of GPU local optimizations. Once we go that, we take a step up in the scaling hierarchy, and the question is, how do we increase the performance of a single trainer? So the model learned best with a number with 512 samples per mini batch, but that was not the most computationally efficient data point. And so we were able to scale up to a mini batch size of 4,096 per trainer. And again, because we're not aggregating we're not doing data parallelism between trainers, that's actually the global mini batch size. If you go farther than that, actually, it stopped converging as well. We tried a lot of learning rate scaling techniques. None of them actually made the model behave better. So this was the peak mini batch size that we could work with. And we found that the most GPUs sampled per GPU you could have is 512 when you're using CUDA graphs. So we were balancing between 256 to 512 samples per GPU to keep the GPUs fed, which meant we had mini batch sizes between 2K and 4K, operating on 8 to 16 GPUs, which turned into 2 to 4 compute nodes on the Sierra architecture. So that's 4 V100s per compute node. We see here in the bottom right that with 8 and 16 GPUs with the different mini batch sizes, we're getting pretty good strong scaling, about 75% parallel efficiency. We could scale it farther, but we chose not to because we started to lose parallel efficiency, and we had more tricks in the hat. So now the question is, how do we train at scale? So we sort of exhausted the local trainer parallelism. So now we're going to scale up to the tournament algorithm that's going to allow us to work with 1.6 billion compounds. And before I go into the technique here, I want to say, let's look at the top table on the right, which is a measure of the test reconstruction error versus the domain-specific result of average Tanimoto distance. So what we found was if we trained the models to a 0.024 reconstruction error, the compounds generated were sufficiently valid. 77% was enough to keep our domain scientists happy. And the tandem distance of the test data set was actually better than the junction tree variational autoencoder. So that was good enough. So it's really nice to actually correlate a reconstruction error to a domain specific metric where the domain scientist gave us a thumbs up, said that is a converged enough model. So for this example of scaling, we are going to train every model to about 0.024 or 0.025 reconstruction error. You see on the right-hand side, the number of nodes going from 64 compute nodes to 4,160, which is the largest we could get in our allocation. And the epoch time is shown on the y-axis. And so what you see is that as we add more trainers, there's basically either 8 GPUs per trainer or 16. So that's 2 compute nodes or 4. So as we go to the right, we're adding more and more trainers. We see 2 things. One, the epoch time gets better because the data set is partitioned over more trainers. So that's good. We get the normal strong scaling you would expect there. The models all converge, but many of them take more epochs, which really is an indicator that because the epochs are all smaller, they're taking roughly about the same number of steps in SGD updates to get to a converged model. But it's important to note that they all do converge. And you can also see that going from 8 GPUs per trainer to 16, we lose a little bit of performance, but we converge in fewer epochs. And again, there's more data per epoch. So there's a design space to explore here. Okay. Now here's sort of the bottom line, which is if we start looking at the time to solution. So on the x-axis here on the top right, it's the total wall clock runtime from when we hit submit to, once the job is queued to when we get an answer back. The y-axis is the test error, so lower is better. And each line is a plot of how many compute nodes we are using, going from 64 at the far right to 4,160. If we look at the far right, we see that with 64 nodes, we can train our model on 1.6 billion samples in anywhere from under an hour to going up to about 2,400 seconds. And if we go from 64 compute nodes to 256 compute nodes, this black line shown here, we get about a 1.8x speed up in the time to solution. So a parallel efficiency of 44%. So that's pretty good. It's important to note that this is now training at 256 nodes, the model to a expected or an acceptable reconstruction error of 0.025 in 32 minutes. 3 orders of magnitude more data than the junction tree variational autoencoder in 32 minutes versus 24 hours. So the ability for our researchers to test new models, new architectures, explore hypotheses is now in human time. It's not compute limited. Now we push this all the way as far as we can because this is a Gordon Bell. And so we push to 4,160 compute nodes that's shown here on the right-hand plot. The model still converged. It still did so faster, but the rate of learning was degraded in terms of how fast we got there. So we got a 2.4x speed up of 4,160 nodes versus 64 nodes. So it achieved 318 petaflops, which is fantastic. But our parallel efficiency was about 3.68%. So we've over decomposed the problem here, but we were able to do it. And a lot of this is actually an indication of the model itself and how it learns at this scale, not the underlying computer science techniques that allow us to do that. So we were able to achieve 17% efficiency of Sierra in mixed precision and get 318 petaflops with these runs. Just to start wrapping up here, there are some interesting lessons learned. When you take the entire, this was the third fastest supercomputer or second fastest supercomputer in the world at the time, and you, third fastest, behind Fugaku and Summit, and you do it to train a single task, you can get interesting calls like the local power company calling up Livermore Computing going, what the heck are you doing? We're seeing 3 to 4 megawatt swings or 2 to 3 megawatt swings, like very fast. Coming from your center. What's going on? Well, it turns out if you look at the wall power shown here in these 2 plots, the left hand is our original algorithm where we're seeing 200 kilowatt power swings per row, and there's 20 racks in each row. at very high frequencies, it turns out that was when we went from training full tilt to when we entered our asynchronous or our LTFB tournament algorithm, where we did 2 rounds of inference and then we spun back up to go to training at full tilt. So we said, let's think about this. So we added a little bit more asynchronous details to the LTFB algorithm. We removed one synchronous point, and that actually got us our right-hand plot where we now got 100 kilowatt swings, 120 kilowatt swings, and it's smoothed out a little bit more. So that was a big improvement. But really what we need to start thinking about is that these leadership class computing exercises can have interesting side effects. Okay. So what I've talked to you about today is our work with the Atom team's Generative Molecular Design loop, and how can we develop new models that can revolutionize the way we do drug design. Fundamentally, we created a model that is much more scalable and efficient to train. We can train using over 1 billion compounds using 16,000 GPUs in 23 minutes. It is significantly better in terms of chemical similarity with regards to the domain-specific measure. I must emphasize at this point that to use the model for inference in this loop does not require leadership class computing. It can be done on any sort of normal GPU or CPU accelerator. So using the model in the GMD loop does not require all these efforts, but training on this large data space does. We are able to use the entire public enamine data library at the time. And the work really was, can we improve the chemical diversity, which is something we're still exploring. But it really allows the domain scientists to start thinking about how do we develop new novel therapeutics for emerging threats going forward in the future. And then from a HPC point of view and a point of view, we have created a toolkit that can use leadership class computing to train neural network models. Getting 17% of peak mixed precision, those 318 petaflops, without loss of the quality of the model when we go to this scale. And that's a big deal. It's done by composing multiple levels of parallelism. And the biggest part about this is it allows you to have a new paradigm for how you do neural architecture search and design, where the time to get insight is in minutes and hours, not days to weeks. And so just one last anecdotal thing is, right before the submission, 2 weeks before, we were working with the variational autoencoder. It wasn't working well. We actually developed the Wasserstein autoencoder in 2 human weeks, reran all the experiments, and were able to retrain at scale in a 2-week design period. And most of that was the human creating new model architectures. So I think this is really unprecedented in terms of how we develop new neural networks. So with that, thank you very much.
Thank you, Brian. I do have one question. Was the failure of training to converge with FP16 due to the small exponent size or small mantissa size of FP16?
We have not done that analysis yet. So there are some tricks in the community about doing better handling of both the mantissa and the exponent that we intend to implement and apply, but we haven't gotten to that, and we haven't had a chance to do that analysis.
Thank you, Brian. Appreciate the presentation.
Thank you very much.